Chapter 11 is on viruses. In this chapter, we will talk about what viruses are and also how they replicate. We'll also be sprinkling in some viral diseases. Remember, this is not a human health and disease class, so you don't have to get too caught up with a lot of the signs and symptoms and treatments. This is really for recognition. So, what is a virus? So, by and large, scientists do not consider viruses to be living things. They are not cells. They do not undergo the characteristics of living things. They don't undergo metabolism. They can't maintain homeostasis. They're obligate intracellular parasites. So, in order to replicate, in fact, we reduce, but in order for a virus to replicate, it has to take over a host cell machinery to make all of its parts. So it's not able to do that on its own without a cell. Viruses have a nucleic acid core, so they are either RNA or DNA, not both. So that nucleic acid, it could be single stranded, or it could be double stranded, it could be circular, or it could be linear. Thanks to molecular techniques, now it's easier to designate viruses as to whether or not they're RNA or DNA viruses. So viruses are going to invade a host cell and they're going to take over that host cell machinery to replicate. As far as vocabulary goes, you will probably hear me go back and forth between calling it a virus, a virion, virions. A virion is basically one viral particle. I talked about the nucleic acid core being either RNA or DNA. And then around that nucleic acid, there is a protein capsid, and this actually protects the nucleic acid. Some viruses have an envelope, not all viruses have an envelope, and that envelope is made of bilipid layers. And our cells have lipid bilayers on our cell membrane. So that envelope is actually stolen from the host cell as those virion particles escape. So as they lyse the cell, blow open the cell, they basically wrap themselves in that cell membrane, and that becomes an envelope. Not all viruses have envelopes, and we'll talk a little bit about some of the advantages and disadvantages of having an envelope. So this is a picture of a generic viral structure. So on the inside, you can see that there's the nucleic acid. This happens to have a DNA core, so this is a DNA virus. And then if you see that purple that surrounds that DNA, that's the capsid. And that capsid is a protein coat that protects that DNA. If you notice that that capsid has all of those different shapes, it almost looks like a little hexagon shape. Those are capsomeres, and capsomeres are those units that actually make up the capsid. We'll talk a little bit about those capsomeres as far as attaching to a cell. A virus is not going to be able to infect a cell unless it can actually attach. We call that attachment adsorption, not absorption, like a paper towel would absorb water. This is adsorption. And the shapes of those capsomeres actually help that virion bind to a cell and to receptors and parts of the cell membrane. If you take a look at this picture, there's also an envelope around this virus. And so you can see those little clothespin-like shapes. That's that lipid bilayer, so those phospholipids. So as this virus escaped from a host cell, it wrapped itself in that membrane. You'll also notice that there are spike proteins. You can see all of those little blue spikes coming off of that envelope. That again is going to help that virus attach or adsorb to a cell. So those spike proteins would actually attach to receptors and allow that virus to adsorb. If that virus can't adsorb and attach to a cell, then it's not going to be able to replicate. If it kind of comes in like a balloon and bounces off, then it's not going to be able to replicate. So, a little bit more about the capsid. So, this is going to be a protein coat that protects that viral genome. Remember, it's either RNA or DNA, not both. And it's made up of those little subunits, those capsomeres. And I talked a little bit about those shapes of the capsomeres helping to adsorb to a cell. This capsid, this protein coat, can actually determine the shape of the virus. So viruses actually have different shapes. We're going to talk a little bit later on sort of the taxonomy of viruses. There's different ways that we can actually classify and kind of put viruses in groups. Sometimes they can actually be grouped depending on the arrangement of those proteins that are in the capsid. So I mentioned a little bit about that envelope. So that generic viral structure that we looked at, that particular virus had an envelope. If it has an envelope, we call it an enveloped virus. If it is naked and does not have an envelope, that would be non-enveloped virus. So because that envelope is made up of phospholipids, proteins, and carbohydrates, like our cells have that cell membrane, it actually can leave them a little more vulnerable to the environment. So they can actually be a little more temperature sensitive, chemical sensitive if they have an envelope, just like our cells would be. So that's why naked viruses without that envelope actually are a little more virulent, meaning that they can actually cause more harm and they can actually last on surfaces a little longer. The one advantage to having an envelope is that having that envelope allows the virus to actually get into cells. So with that envelope, it kind of melds with the plasma membrane and it allows it to get in because it also is enveloped with this membrane, sort of this plasma membrane like coat. It can actually hide from the immune system. So it's almost like this cloak of invisibility from the immune system so that the immune system doesn't recognize that it's a virus. So the advantage is the envelope allows them to gain access to cells a little more easily, and it can go a little bit undetected by the immune system. However, because it's going to be prone to different environmental conditions, it can make it a little vulnerable to changes with like pH and chemicals and things that would harm and get rid of that virus. So how big are viral particles? So if you take a look at this picture, the peach part is actually a human liver cell. So that would be a eukaryotic cell. And then the green cell, and you can actually see the flagella around the surface, that's E. coli. So that is a bacteria. So we know that bacteria are much smaller than our cells. And then if you see all of the different shapes of the viruses and the sizes, so you can see the picornavirus is very tiny compared to like the herpes virus. You can also see the tailed bacteriophage, so you can get an idea of how big these viruses are. So remember, we would not be able to visualize a virus underneath our compound light microscopes. We would actually need an electron microscope to visualize a virion. So I mentioned that capsomere can help determine the shape of viruses. So viruses can come in a variety of shapes. Some can actually be helical, like the tobacco mosaic virus, which is one of the most studied viruses out there. Some can have almost like a geometric shape where they can have many sides, like the adenovirus or adenovirus, however you'd like to say it. Some can kind of have like different helical shapes, like our tailed bacteriophage. I'm going to show you a pictures of a rabies virus that looks like a bullet shape, and Ebola virus that looks like a thread. It almost looks like a worm. So here are some different viral shapes. So you can see rabies virus kind of looks like almost like a cylinder, like a bullet. You can see the adenovirus or adenovirus. where it looks geometric. Of course, it looks two-dimensional, but three-dimensional, it would probably have more sides to it. There's the tobacco mosaic virus, where you can kind of see that helical shape. And if you look on the bottom left side, you can see Ebola. And I remember when this was in the news several years ago, on the news, they would have pictures of Ebola. It almost looks like a parasite, like a worm, because of that thread-like configuration. So viruses can come in a variety of different shapes. So, a little bit more on rabies. So, within this chapter, I've tried to sprinkle in some diseases just to make it a little interesting, because I know that's probably your point of reference and also interest as well. So, rabies, as we know, can enter through animal bites. So, this has to come from direct contact. This can then travel to the nerves, and then it travels to the central nervous system, the brain, and spinal cord. Symptoms and the incubation time of rabies can vary, where people can get headaches, fever, nausea, partial paralysis. Appearance of symptoms can vary. I mean, again, the incubation time can be anywhere from 13 days to 2 years. If you Google rabies, you definitely do not want to wait the 2 years. And it's usually recommended that if you've been in contact with an animal that you suspect has rabies, even if you cannot actually have somebody capture that animal and test it, that you should probably be getting that rabies series, and it's a whole series. Every once in a while, you hear in the news about somebody being attacked by a rabid animal. I remember in the news a few years ago, there was a woman and she saw a dog in the parking lot that she thought was in trouble. And so she went to rescue it. Turns out it was a rabid raccoon that attacked her. And of course, she had to go for the rabies series. Recently, last summer, I have a friend. She works in New York City, but she lives in Jersey City. And she rescues many cats, but her neighbor had actually found a stray. It was a little kitten, and she was coming home from work, and the neighbor was outside with a kitten, and she went over, approached her neighbor, and the kitten attacked her, and she was scratched up. And so it took a while to get the testing done. The kitten did have rabies. She did not hesitate. Even before they tested, she went to the doctor to get that series. And of course, she was putting out on social media, just be forewarned, be very careful of strays and wild animals. So, what do viruses infect? Viruses are going to have a host range. So, most viruses are only going to infect one type of species, a plant or a bacteria. As I mentioned with the bacteriophage, that is not a virus that can actually attack our cells. An example could be poliovirus. Polio is only going to cause disease in humans. We would not see this in other animals. Of course, we do know that there are some viruses like rabies that can actually infect a variety of species. And so the variety of species and what that virus can actually absorb and take over with its replication process has to do with its host range. Viruses need to be very specific because of that attachment. So in order for it to replicate, it has to adsorb. So if the receptors on that host cell do not bind with those capsomeres or those spike proteins, then it's not going to be able to adsorb. So those receptor sites are going to be very specific. So that's why viruses sometimes are very specific with the species that they will infect. The other thing is that the enzymes, the host machinery, has to be compatible with the virus. If that virus is able to enter into the cell, but it can't take over that host cell machinery, then it's not going to be able to replicate. It's kind of like if you've never flown a plane before and you get into the cockpit of a plane and you're looking at all of those dials and buttons and you're going, there's no way that I'm going to be able to fly this. That's that incompatibility. So if that virus can enter, even if it can absorb, if that host machinery is not compatible, it's not going to replicate. That's what we would call an abortive virus. Yes, it can get in, but it's not going to be able to replicate. We also know that viruses will infect certain tissues. So, papillomavirus can only infect skin cells. That causes warts. And we'll talk a little bit more about papillomavirus. Cytomegalovirus, CMV. This is something that the majority of us have probably been exposed to. And unless you're immunocompromised, it probably hasn't caused too much of an issue. But this type of virus can actually infect a variety of tissues. And so it can present in different ways and cause varied symptoms that sometimes makes it a little difficult to diagnose. So some viruses are very specific with their tissues, others may actually invade different tissue types. So, because we talk about viruses not being living things, the question that always arises is, where did viruses come from? And I think the jury is still out. That some scientists say, well, maybe at one time they were real cells and now they're just pieces of cells. Maybe they came from bacterial plasmids. In looking at viral evolution and how viruses arise, many scientists believe that maybe they came on the scene at the same time as cells, since they require host cells to replicate. So, this is something that is ongoing, and certainly with certain viral types, there is a lot of study into viral evolution. We certainly have done a lot of research in the past few years over COVID. And so with the coronavirus, they've been studying how it's changed and how it's evolved. Viruses will usually evolve as they're passed through populations. And that's how those changes and mutations can actually occur. So, how we actually classify viruses is a little different than how we classify living things. So, if you remember, we talked about binomial nomenclature and we also talked about the classification system. Originally, it was based on the type of host a virus could infect. So, whether or not it was like an animal virus or a plant virus. They could also classify them depending on what type of area, what type of tissue was actually affected. So, did it affect nervous tissue? The naming of viruses is a little different than the naming system that we talked about with binomial nomenclature. Mostly, we use the common name for the species. So, something like poliovirus or rabies virus. Because of molecular techniques, it's now we can actually classify and put viruses in their categories of being either an RNA virus or a DNA virus. So, we are going to be getting into some different RNA viruses. So, we'll talk of some examples, some I'm sure you've heard of, and DNA viruses as well. We'll also be talking about how viruses replicate. So, how they absorb, how they take over the host cell machinery. We were talking a little bit about the evolution of viruses. Remember, viruses can actually change over time. As they pass through populations, they can change. And it's also not an advantage for viruses to kill their host, because then that means that they can't replicate. So, success in terms of a virus is to be able to get in, replicate, and continue on with that viral population so it can continue to infect cells. So, we will be talking about all of those things as we go through this chapter.